everybody. Welcome to Chapter 5. In Chapter 5, we're going to be talking a little bit about the integumentary system. So we're moving away from talking about the smaller aspects of the human body when we were talking about cells and how cells combine to make tissues, and kind of moving on to the larger systems itself. So our first major system that we're going to talk about is the integumentary system. Now, the integumentary system consists of several things. Mainly, you can refer to it as the skin, but basically there are several parts of the integumentary system. Like I said before, things like the skin is in part of the integumentary system, but also hair, nails, sweat glands, including sebaceous oil glands as well. So we're kind of going to go through each of these, talking a little bit about not only the cells, but the tissues that make up this entire system and each parts of that system. So first we'll talk about the structure of the skin. So the skin does have these two distinct regions associated with them. So there's the epidermis and the dermis. So the epidermis is that superficial region. It sort of connects the epithelial tissue, and it's avascular. The dermis, however, it underlies that epidermis. So it's mostly that fibrous connective tissue we talked about back in Chapter 4, and it is vascular. Now, we do also have a hypodermis, and that's what we call the superficial fascia. So it's subcutaneous layer deep to the skin. Remember our body positions, but it's deep to the skin. Not part of the skin, which is why you heard me say we have two distinct groups, epidermis and dermis, but we also have this it's not a third group, but this third thing that we're talking about, which is not part of the skin, but it does share some functions with that. The thing is, the reason it is not really part of the skin itself is because it is mostly adipose tissue. So adipose tissue, of course, is fat tissue, and that basically is the part that's going to help absorb shock and helps insulate, keeping us warm. It also anchors the skin to these underlying structures. Mostly the muscles that are there is what that hypodermis is going to anchor to. So adipose tissue with some anchoring the skin to those muscles as well. So here's an image of that skin structure. So you can kind of see this epidermis layer that we talked about, pretty small in comparison to that dermis layer that we see here. So you kind of see hair follicles, you see veins and arterioles and venules and things like that. You see nervous tissue as well. And then you have this subcutaneous, this hypodermis, which again is not part of the skin, but has some functions that helps protect us in that. So you can kind of see those fat cells, that adipose tissue that's laid down there. And of course, that's going to connect that to that muscle. So the epidermis in general basically consists of mostly keratinized stratified squamous epithelium that we talked about. We were talking about different types of epithelial tissue, and there are four types that are found in the epidermis. So the first one of the cell types that we'll talk about is the keratinocytes. So the keratinocytes, they produce this fibrous keratin. It's a protein that sort of gives that skin its protective property. So several things that we have on our body has keratin in it. It's fibrous and sort of helps provide that barrier, allow it to be protected. There are major cells of the epidermis, so it's why we're talking about it first, because it's that biggest one there. And it's tightly connected by those desmosomes. And if you remember back in Chapter 3, we talked a little bit about the connections between cells side by side. And so they are tightly connected by desmosomes, making those one kind of unit so that they can stick together. Now, with regards to the keratinocytes, millions of these slough off every day. So if you reach your arm out and take your left hand on your right arm and just kind of rub back and forth, and just millions of little skin cells are falling off. These are mostly these keratinocytes. So that's what's coming off when we slough off our skin or lose some of our skin. Then we also have the melanocytes, and these are sort of these spider-shaped cells, and they're really located deep in that epidermis. They do produce the pigment melanin, which is packaged into these little melanosomes. And basically, melanosomes are transferred to those keratinocytes where they protect a nucleus from UV damage because the nucleus, remember, contains our genetic material, our DNA, and sun can damage that, causing mutations and different things like that. So we want to protect it from the sun, and melanin does that for us. There's also some dendritic cells. These are also called the cells of Langerhans. They're star-shaped macrophages that sort of patrol deep in the epidermis. And I know we haven't talked about much about what macrophages do, but basically they're those cells that go and eat debris and all kinds of stuff that you don't want there. If you have an infection, if you have a cell that's dying and they need to sort of be broken down, taken apart, and disposed of, these macrophages will do that. They're cell eaters and debris eaters. So they are the key activators of the immune system. So when a macrophage comes in contact with something that's dangerous to us or harmful to us, those are the ones that are going to part of the response to trigger that immune system to say, uh-oh, something's wrong. We need to take care of this. We also have some tactile cells. These are also called Merkel cells. And these are sensory receptors that sort of sense touch. So, one of the things that's important is that we can respond to stimuli. So basically we have these Merkel cells that if we touch something, we can feel that sensation, whether it's a pleasure sensation or pain sensation or, just so that we can sense that. And those help us because through these Merkel cells to feel things and sense things. Now, the epidermis itself is made up of basically what we call 4 or 5 distinct layers. So if you have this thick skin area, basically you have 5 layers or strata. So if you hear me say layers or you hear me say strata, it's the same thing. But thick skin have the 5 distinct layers, and other thin skin only contains the 4 layers or 4 strata. Now, thick skin is found in areas where it's super high abrasion areas, hands and feet, things like that. But then you have thin skin pretty much everywhere else. So these 5 layers of skin, and you'll see that basically, and I'll call off what will be in those 5 layers of thick skin. You have the stratum basale, the stratum spinosum, the stratum granulosum, the stratum lucidum, and the stratum corneum. Now, in thin skin, you do not have the stratum lucidum. So that's only in thick skin. So traditionally in most areas of our body, we're going to have that basal, spinosum, granulosum, and corneum. But in our hands and feet and things like that, we're going to have that extra layer in that stratum lucidum. So the stratum basal is the basal layer. That's the deepest of all those epidermal layers. It's also called the base layer. That's the layer that is firmly attached to the dermis. And so basically it consists of this single row of stem cells that just divide via mitotic division, producing 2 daughter cells each time. So one daughter cell journeys from that basal layer, that deep layer, up to the surface of the skin. Now, it takes some time to do this. It doesn't do it automatically. But basically what happens is, as that daughter cell divides from the stem cells, it starts working its way up. And basically till it gets to the surface. That takes about 25 to 45 days to reach the surface. The way it's getting there, the cell itself isn't necessarily, I'm trying to think how to word it, isn't necessarily actively moving up per se. I mean, yeah, it is a little bit, but also the fact that you're losing some of those dead cells that are sloughing off as well. So as cells die and slough off, you start seeing these new daughter cells journey to the top. They eventually will, of course, die off, and new cells will move up and replace those. And so that's about 25 to 45 days for it to reach that cell surface. So our skin regenerates. While they give you a range of 25 to 45 days, I always just say an average of a month for your skin cells to regenerate. This is why you can have, I'm trying to think of an example. If you've taken a chemistry class and you've worked in a lab with certain chemicals or dyes and it gets on your skin, your professor may say, don't get this on your skin because it won't wash off, and you just have to wait for your skin cells to regenerate. And usually they'll say it'll take about a month, a little over a month. And so that's what they're talking about. It's that stain gets in there, and then of course over time, it can wear off. So the cells, as they die, these move to the surface. Their other daughter cells remain in the stratum as a stem cell. So like we talked about, there's those two cells that are produced. One will continue to work its way up, but that other one remains as a stem cell so that it can continue to regenerate those every month or so. Or at least to the top. It regenerates them all the time. But the layer is also known as the stratum germatavium, and that's because of the active mitosis. Germ means, like a germ cell, the original cell that's going to divide and create, more cells. Remember, mitosis is the division of those cells, so that's why they call it the germatavium. So another thing that's composed in this stratum basal is 10 to 25% is composed of melanocytes. And so those are the types of cells and things that you're going to find there. In the stratum spinosum, the word spinosum, it's called that because it's also called the prickly layer of the skin. There's several layers thick, and they kind of have this little web-like system of this intermediate pre-keratin filaments that are attached to those desmosomes. So what these do is it allows you to sort of resist tension and pulling. So if you needed some of these cells to pull around, if you think you can stretch your skin a little bit, you don't want it to rip, right? You want it to be able to sort of be able to be pulled some and to be able to be stretched some. And so that's something that this layer can help with. The keratinocytes in this layer also appear spiky, so we call them sometimes prickle cells. So if you hear it referred to, like in a textbook, as prickle cells, they're also referring to the keratinocytes. These are scattered among these keratinocytes are lots of melanosomes and dendritic cells as well. So lots of different things going on in that stratum spinosum. In the stratum granulosum, that granular layer, it's 4 to 6 cells thick, but they're kind of flattened, so they're very, very thin. So the cell appearance does change. So cells flatten, and of course this nuclei and the organelles can disintegrate, and this keratinization begins. So cells accumulate these little granules that help form these keratin fibers in the upper layers. They also accumulate what we call lamellar granules. It's this water-resistant glycolipid that helps slow water loss. You can think of that. That's important, right? We need a lot of water in our system. It helps to keep us alive, keeps our body going, keeps metabolism functioning as it's needed. So we have pores in our skin, and we don't want the water just to leak out all the time, right? We want to be able to control it and contain it. So these little lamellar granules have this glycolipid. It's just a lipid with this little glycan attachment to the head of that lipid that helps keep the water in. Remember, lipids are don't like water, so they're hydrophobic. So basically, it's going to keep that water on one side of it. And then, of course, cells above this layer here, the stratum granulosum, basically die. So it's too far from the dermal capillaries to survive. That's why our skin starts sloughing off. There's not a whole lot at the top of these capillaries to provide the blood supply to these upper layers. So over time, they die off and slough off, and it continuously replaces itself. Now, there's a stratum lucidum. It's a clear layer. It's only, like we talked about before, found in thick skin like your hands and your feet. And this consists of this thin, translucent band of 2 to 3 rows of these dead keratinocytes. This lies superficial to the stratum granulosum. And then we have this stratum corneum. We call this the horny layer. It's basically 20 to 30 rows of these little flat, anucleated, keratinized dead cells. And basically, this is about 3/4 of the epidermal thickness. Now, although these are mostly dead cells, they still have a function for us. They're very, very important. One, they protect these deeper cells from the environment. So if your cells on the top of your skin, are already dead and there's some kind of sun damage hitting those, top layers, it stops that UV light. It slows down that UV light from getting deeper. So or any other environmental conditions. If we slightly get a paper cut, it's not, it may not be deep enough to hurt the lower layers of our skin. It really protects us from the environment, those deeper cells. It also works to prevent water loss as well. And of course, I've already mentioned this, but protects against from abrasion and penetration. So basically, this layer, the stratum corneum, really is our barrier against biological, chemical, and physical assaults. So the epidermis plays a huge role in really protecting us against the outside world. It is a protective barrier. Now, when we're talking about how these cells change by going through the process of apoptosis. I think I've mentioned this word in a previous chapter already, maybe in a cell chapter. Apoptosis, I think I mentioned that some people say apoptosis, but apoptosis is cell suicide. It's basically this controlled, regulated cell death. We don't want our cells to live, if they could be healthy and no damage and no DNA changes, we would want our cells to live forever. But because our cells age and because they get changes to their DNA structures, we don't want them necessarily to keep living with all the errors that they accumulate. So when they get damaged or anything like that, they're going to kill themselves, essentially. It's a programmed suicide. And so this dead cells slough off. They come off as what we think of as dandruff or dander. And so we basically shed about 50,000 cells every minute. So if you think about, without even touching your skin, really, they're sloughing off. And of course, if you scratch or rub, you're going to lose more of those cells at that time. Here are some of the cells and the layers of that epidermis. So you can see each of these layers, the stratum corneum, see how flattened those cells look. You see deeper down in the granulosum and even lower, you see these keratinocytes. You see all of these other cells that you see down here, all working to sort of protect you. This upper layer we talked about, remember that's 20 to 30 layers of dead cells thick. That's a protective mechanism to protect these lower layers. So we're not damaging those cells, we're not damaging the dermis, we're not doing all of that stuff. So this is really that protective layer. You could see them very clearly in this little flattened image. So since I mentioned the dermis just a second ago as being deep down, dermis is this strong, flexible connective tissue. So there's a lot of different cells in here too. There's the fibroblasts, the macrophages, and of course the mast cells and white blood cells that come along as well. So the fibers in this matrix bind this body together. So it makes up the hide that is used to make leather. So when you think of leather making process, this is what these fibers and this matrix that can bind together. That's what makes leather. It contains nerves and blood vessels, lymphatic vessels, and of course it has the hair follicles. So those epidermal hair follicles, the oil glands, the sweat glands. And there's really two main layers to the dermis, the papillary layer and the reticular layer. And so you can see here, this is the epidermis under a light micrograph, and you can see all the way down to the dermis. Well, here is the papillary dermis. That has some areolar connective tissue associated with it. And then even deeper down, you have this reticular dermis, which is this dense, irregular connective tissue. So these little things right here are that dermal papillae that I mentioned before. So that's sort of under this light micrograph of what that dermis would look like. So that papillary layer, again, like I mentioned before, it's that superficial layer of that areolar tissue, connective tissue, that is sort of this loose, sort interlacing collagen and elastic fibers with some blood vessels. The dermal papillae is the superficial region of the dermis that sends these finger-like projections up into the epidermis. I showed you that just a second ago. You can kind of see that dermal papillae kind of protruding up a little bit. And then, of course, there are some dermal modifications in the skin that does result in these skin markings. So you can think of the ridges on your fingertips making up your fingerprint. Those are basically epidermal ridges that lie on top of dermal ridges. You also have little openings in those, which are sweat duct openings along the crest and of those friction ridges to help make the fingerprints. And so the skin does provide these very unique skin markings, which we use, of course, to identify people, especially in forensic science and things like that. Some other modifications that you can think of, it's not an other modifications, it's similar, but we do have some dermal modifications and skin markings all over our body. We have things called cleavage lines, and they sort of represent the separation between underlying collagen fiber bundles and sort of the reticular dermis. And so they sort of run circularly around the trunk and longitudinally on the limbs. Surgical incisions parallel to these cleavage lines heal better. So when you have a surgery, the doctor is going to try to provide or do a cut on the skin parallel to those cleavage lines so that they heal better and heal with less scarring as well. Of course, we have some other characteristic markings. You can see flexure lines. Those are from where the dermis is closely attached to the underlying structures. So you can see those. They almost look like wrinkles in your hand, but they're not wrinkles. They're bend marks. And, of course, everybody has them, and they're slightly different in everyone as well. Some other things you might see on the skin are things called dermal tears or striae. These are also commonly called stretch marks. So stretch marks or dermal tears happen when the skin starts expanding too quickly, really, for that skin to really produce new cells of skin. And so it forms this sort of scar tissue in placement to keep the skin together so it doesn't rip apart, right? We don't want that to happen. And so basically, you get these little scarrings that happen. The most common place you see these are in women who are pregnant because really their stomach is growing at a really rapid rate. And so these stretch marks form just so that the skin doesn't rip apart. Basically, the skin can't replace itself fast enough, and it's growing too fast. It's not just in pregnant women, though. Men can get them and women can get them just if you're gaining weight too rapidly. So if you gain a lot of weight in a short period of time, you will also see these markings. And so that will appear as well. These commonly appear on the stomach. That tends to be something that comes pretty fast. Some people can get them on their thighs and their buttocks, and some in the arms too, if the upper arms are growing pretty quickly. These do not go away. You can see here there's this purple looking color to it. One thing to say is if they're these purple pinkish looking colors, they're still expanding. And they haven't quite, healing is not the right word because they're going to be scar tissue. But after they stop stretching, they will be more of like this white scar tissue. They do not go away. They also do not tan. So some people think that they can tan their stomach and try to hide them. And no, they do not tan. So your other skin will get darker in color, but the stretch marks will stay white. And so a lot of people with stretch marks tend to try not to get any too much sun, so that hides it the most. Another way to hide it too is if you get an experienced spray tan person that can do it, spray tan really well. They know how to cover them, but you usually have to find a really good one. So I've known people to cover their stretch marks through spray tans because that can cover it. It still leaves a marking so you can see it, but it will be seen less. So There are three pigments, so now we're talking about skin color and those pigments in the skin. There are three pigments that contribute to skin color. The first one is melanin. It's the only pigment actually made in the skin, and it's made by the melanocytes, made from the amino acid tyrosine and tyrosinase. And they're packaged into these little melanosomes, and they're basically sent to shield DNA of keratinocytes from damaging UV light. There are really two forms of that: reddish, kind of yellow, to brownish black. And so skin color differences are due to the amount and form of melanin that are there. Freckles and pigmented moles are also just small local accumulations of melanin. So a lot of our skin color is determined by how much melanin you have in your skin and how much is produced at particular places. The more sun, the more the need for a protective shield, so the more melanin will be produced. And so that's why you basically can get darker skin if you're in the sun more. So one thing, excessive sun exposure, you can think of as being an issue that many people in America, well, the whole world face, but it's something that we talk about in the United States as well. You know, using sunscreen and all of this stuff. Basically, excessive sun exposure damages the skin. So these elastic fibers begin to clump together, and it causes the skin to really become leathery. And so that can depress the immune system and basically damage the DNA, which leads to skin cancer. UV light destroys folic acid. That's necessary for DNA synthesis, so insufficient folic acid is especially dangerous for developing embryos. Photosynthesis Photosensitivity is increased in reaction to the sun. So things like some drugs, antibiotics, antihistamines, even perfumes can cause photosensitivity, and that can lead to damaging, the skin rashes or faster sunburns or things like that, more damage at a faster rate. There's also the pigment carotene. That's this yellow to orange pigment. You mostly see these in palms and soles, but it accumulates in the stratum corneum and the hypodermis, and it can be converted to vitamin A for vision and epidermal health. And then we also have hemoglobin, and so that's that pinkish hue of fair skin that's due to the low levels of melanin. So skin of Caucasians is a little more transparent because there's less melanin there. And so the color of hemoglobin shows through. So someone who is Caucasian can have this pinkish look to them because their skin is a little more transparent due to the lack of melanin. There's some alterations also to think of in skin color that we can indicate disease. There's one called cyanosis. That's that blue skin color. That means you have low oxygenation of hemoglobin. You can have what we call pallor, which is blanching or a very pale color. That can be caused by anemia, low blood pressure, fear, anger, different things like that. There is erythema, which is redness, and that can be caused by fever, hypertension, inflammation, allergies, so on. And there's also jaundice. That's this yellowing of the skin. You can also see the jaundice in someone's eyes when it's really bad as well. And those are indicative of liver disorders. So basically, those are some things, that homeostatic imbalances that we can see just by looking at skin. Also, bruises, those are those black and blue marks that's referred to ecchymosis or hematomas. These are the result of clotted blood beneath the skin. As the clot is broken down, the color of the bruise changes as that hematoma or that clotted blood is coming apart under the skin. And then, of course, these brown or black necklaces or bruises, these are sort of hyperpigmented dark areas in the axillae around the neck, and it may be a sign of insulin resistance or even elevated blood glucose levels. Kind of moving away from the skin, we're going to talk a little bit about the hair. So in the hair, these consist of these dead keratinized cells. There's none really usually located on the palms, the soles, the lips, the nipples, and portions of the external genitalia. But the function is really to warn of insects on skin, that crawling sensation, it causes the hair to raise, so something on the skin. Hair on the head guards against physical trauma. It helps protect from heat loss. It also can shield the skin from sunlight as a protective barrier. Hairs, also called pili, are these flexible strands of dead keratinized cells. They are produced by hair follicles, and it contains this hard keratin, which is a little bit different than that soft keratin we find in the skin. So hard keratin is tougher and more durable, and the cells don't necessarily flake off. That would be bad because we do shed a little bit of hair, but if we just constantly lost 50,000 hair, pieces of hair a day, everybody would be bald, right? There are regions of that hair, the shaft and the root. The shaft extends above the scalp, and that's keratinization is complete. And the root is within the scalp, and keratinization, that stiffening up, that hardening, getting tougher, is still actually going on. There are three parts of the hair shaft: the medulla, the cortex, and the cuticle. The medulla is the central core of the large cells and air spaces. And then there's the cortex, and that's several flattened layers surrounding the medulla. And then the cuticle, that's the outer layer consisting of these overlapping layers of single cells. Hair pigments are made by the melanocytes in the hair follicle, so there's different combinations of melanin. You can have yellow, rust, brown, black, and they all create these hair colors. Red hair is unique because it has an additional pigment that other hair doesn't, and that is pheomelanin, and that will result in the red hair. Gray and white hair results when the melanin production decreases and little air bubbles sort of replace the melanin in the shaft, and it kind of creates this gray-white look to it. So red hair and gray hair are sort of abnormal, and I don't want to say abnormal in a bad way, but different from those typical melanins that we see of other hair colors. This is that structure of the hair follicle. So this is a cross-section, sort of a cartoon image of the cross-section, and here it is under a photomicrograph. And so you can see that there's this follicle wall. You have some peripheral connective tissue, this little glassy membrane that's around it, and then you have this epithelial root sheath, the external root that goes around it, and the internal root sheath with the cuticle located there. And so you kind of see the different layers that make up each one of those hairs. This is sort of a structure of the hair follicle itself, so you can kind of see that longitudinal view of that expanded bulb of the follicle. You see these adipose tissues that's in that subcutaneous levels. Cartoon image here and the photomicrograph here, kind of showing you the different aspects of that with the medulla and the cortex and the cuticle of that hair root, the melanocytes that are located there, the hair papilla, and the hair matrix as well. There are types and growth of hair. We have something called vellus hair. That's the pale, fine body hair of children and adult females. And then, of course, terminal hair. And those are coarse, long hairs. They're found on the scalp, the eyebrows. At puberty, they start appearing in axillary and pubic regions of both males and females, and also on the face and neck of males as well. Nutrition and hormones do affect hair growth. So depending on your nutrition and depending on your levels of hormones, you can have more or less hair loss, hair growth, and so on. Now, follicles cycle between active and regressive phases. So basically, you grow about 2.25 millimeters of growth per week, and you lose about 90 scalp hairs daily. So it's normal to lose hair. Men tend to start going bald over a certain time. Some, not all. Women, there is women who lose hair as well, especially as their hormones change through age and towards menopause. But so nutrition is very, very important for hair growth and nail growth, which we haven't talked much about nails, but skin and stuff like that. So making sure hormones and nutrition is, on point as much as possible. Hair thinning, I mentioned a little bit about that. One is alopecia. That's hair thinning in both sexes after the age of 40. However, people can have stress-related alopecia at any age. So there can be, somebody in their teens and 20s going through extreme stress or something like that and can be experiencing patches of hair loss too. Um, Usually, as you age, it's sort of an overall hair thinning of alopecia as opposed to stress, which can come out in more clump-like areas. And then there's of course true what we call frank baldness, and that's sort of genetically determined. It is a sex influence condition. There is, we call that male pattern baldness. It's basically caused by a follicular response to DHT, which is dihydrotestosterone, and that basically causes men to go bald. Men's baldness can start in their teens, depending on their genetics, and lose their hair really early on. It could be losing overall, or it could be receding hairline to have the top of the head kind of bald as well. Now, talking about the structure of the nail, that is also made of a hardened keratin as well. You can see sort of this section here. You see this sort of bone that comes up underneath that nail. You see blood cells. You see this nail fold right at the base of the nail there. And, of course, the free edge of the nail and the nail plate. There is a nail matrix where that nail will grow outward from. And of course, these are just some of the glands that we're going to be talking about. There's some American glands that we'll talk about. It's a type of cutaneous gland. And what you see here is those glands here's the hair follicle. You have the sebaceous gland that will release oils. You see that duct located here and some secretory cells so it can excrete that. There is also a gland here that will allow sweat to come out as well in this eccrine gland, the American gland. The sebaceous gland, of course, too, I mentioned that in the last slide. You can kind of see this is that hair in the follicle, and this is that sebaceous gland duct where that oil can come out. So you can kind of see what that looks like under a microscope there. So this is just a summary of those cutaneous glands, the eccrine sweat gland that we have, mostly for temperature control, some antibacterial properties as well. The apocrine sweat glands, these may act as sort of the sexual scent gland. There's a filtrate. It's a type of secretion, the blood plasma with added proteins and fatty substances. And then, of course, you have sebaceous. It does a couple of things. It lubricates the skin and the hair. It helps prevent water loss, and it has antimicrobial properties to help protect so that stuff doesn't get into the system. Cradle cap is something that you might see in newborns. It's called seborrhea. This can happen in adults. We don't call it cradle cap in adults, but this is interesting because you see how this has this little crusty, like whitish, yellowish. It looks like kind of scab-like. It's not a scab. It's an secretion that's drying. That's really hard to get off. You can't scrub it off very easily. Doctors and scientists still disagree on what caused it. It's not a bacterial infection. It's not contagious, but it is thought to be due to overactive sebaceous glands. That's why it's called seborrhea. And they think it's overactive, it excretes, and then it creates this hardened-like substance there. Some doctors have said, though, it has been seen with some yeast infection up there, but most doctors and scientists will say, while we don't know 100% what causes cradle cap in newborns, it is thought just to be this overactive sebaceous gland. And pretty much it will go away over time. So we've talked about the structure of the skin and all that, and although I've mentioned some functions, we're going to talk a little bit about functions here. So skin is the first and foremost a barrier. That's its main function. So its main functions include protection, that barrier that I just mentioned, body temperature regulation, cutaneous sensations, feeling, so that you can sense, heat. If you touch something, if it hurts or something pokes your skin, you can feel it. It serves for metabolic functions. It is a blood reservoir as well, and it's part of helping you excrete wastes out of the body. So that's some of the functions. So it does a lot for us, right? So for protection, of course, skin is exposed to a lot of stuff: microorganisms, abrasions, temperature extremes, harmful chemicals. So basically, this makes up sort of it's a barrier in three ways, right? It's a chemical barrier, it's a physical barrier, and it's a biological barrier. So it does a lot of work for us. Again, chemical barrier. The skin secretes chemicals like sweat, sebum, defensins to kill off bacterial infections or microbial infections. Um, Acid mantle is also part of that. It basically the low pH of skin sort of retards or basically stops or slows, I should say, bacterial multiplication. So if you get a ton of, I don't know, like E. coli on your hand, right? Obviously that's bad. But the low pH of the skin, bacteria need a certain pH to grow in. And if your skin is a lower pH than that bacteria that's on your skin, example E. coli, it can slow its growth or stop its growth. More than often than not, it's just going to slow its growth and not stop it. But that's the acid mantle, which really helps defend against pathogens. And then melanin provides the chemical barrier against UV radiation damage. And then, of course, I mentioned the physical barrier a minute ago. Flat, dead, keratinized cells of this stratum corneum. It's surrounded by glycolipids. Some chemicals do have limited penetration to skin, things like lipid-soluble substances, plant oleosteins, that's things like poison ivy. Some organic solvents like acetone and paint thinner, salts of heavy metals like lead and mercury, drugs like nitroglycerin, and other drug agents which help carry drugs across the skin. So some things can go across, but most things can. It serves as a barrier for most things. So then again, we've mentioned this already, but basically these biological barriers, that there's some phagocytic cells there to break up any invaders that come in. Dendritic cells of the epidermis will engulf invaders and present them to the white blood cells, which activates this immune response. So then it kind of protects us, right? And then, of course, this dermis contains those macrophages I mentioned before. They activate the immune system by presenting these foreign antigens to the white blood cells. And then DNA can absorb this harmful UV radiation, converting it to this harmless heat. It's also really important in body temperature regulation, of course, and so body temperature regulation is important with regards to the skin. So under normal temperature, sweat glands produce about 500 milliliters per day. We call this insensible perspiration. But when body temperature rises, the dilation of dermal vessels can increase sweat gland activity to produce 12 liters, which is about 3 gallons of noticeable sweat. We call this sensible perspiration, and that's designed to cool the body. We call this this cooling can be referred to as evaporative cooling as well. You could, even when you don't sweat, you can cause evaporative cooling by putting water on you as well. But in the cold environment, the dermal vessels start to constrict and the skin temperature drops to slow passive heat loss. So continuing on, basically a little bit about the metabolic functions. Skin can synthesize vitamin D needed for calcium absorption in the intestine. And so that's why it's important when it comes to calcium absorption that you also have vitamin D synthesis. The best way to undergo vitamin D synthesis is to get out in the sun, let the sun hit your skin. And so when you consume calcium, it doesn't get absorbed unless you synthesize vitamin D. Now, some people can't be out in the sun. Maybe they live in extreme northern or southern hemispheres, and their daylight is, shortened or different times of the year, or just somebody likes to be inside. And so now if you buy things like milk or something like that, it will have the calcium, but it'll also have plus vitamin D. You can see that in orange juice as well. And that's to make sure people get the vitamin D so that the calcium actually absorbs. Because calcium is essential not only for strong bones, but also for intercellular messaging between cells and communication. Chemicals from keratinocytes can disarm some carcinogens. And also those keratinocytes can activate some hormones, such as converting cortisone to hydrocortisone. And then, of course, the skin makes collagenase, and that's which aids in the natural turnover of collagen to prevent wrinkles. The skin serves as a blood reservoir, which can hold up to 5% of the total blood volume of the body. And of course, skin vessels can be constricted to shunt blood to other organs such as when exercising muscle. And of course, excretion. Skin can excrete limited amounts of nitrogenous waste, such as ammonia, urea, and uric acid. Sweating can cause salt and water loss too. That's why it's important to replenish those things, especially water. And with regards to skin cancer, the skin can really develop over 1,000 different skin conditions and ailments, and many internal diseases can actually reveal themselves on the outside of the skin. Way early in the chapter, I was talking about pigments and how sometimes we can see pigments produced that will change the color of our skin, and this can indicate bad news, like jaundice. Jaundice causes the yellowing of the skin, and that the outside of your skin and even your eyeballs can look yellow, and that kind of tells us that there's a liver condition. So you can see your skin change on the outside in their color, and that could indicate some kind of issue on the inside, such as jaundice and liver disease. Some most common of these disorders mostly are infections. So the skin tends to have, if it gets past the barrier, we get a lot of infections. But less common but more damaging are things like skin cancer and burns. Most skin tumors are benign that are not cancerous. So, they begin as this little tiny growth and they don't necessarily spread or metastasize. So these risk factors are things like overexposure to UV radiation, frequent irritation of skin, and so on. Some skin lotions contain enzymes that can actually repair damaged DNA. But there are three main types of skin cancer. There's basal cell carcinoma, squamous cell carcinoma, and melanoma, and we'll talk about these. Basal cell carcinoma, of course, is the least malignant and most common. The stratum basal cells proliferate and slowly invade the dermis and the hypodermis, and this is cured by surgical excision in 99% of the cases. This can be referred to as basically this surgical excision can be referred to as the Mohs procedure. So you may have seen this in your grandfather or someone in your family having a surgical excision, and they may have referred to it as the Mohs procedure, and that's sort of what that is called. This is a photograph of basal cell carcinoma. This is a rather large one on a man. They can look just like zits. So the one way to distinguish them, if you don't know how to distinguish them by feeling them, they do not heal. So like if a man shaves his face or it breaks open and it doesn't heal, it may be a sign that you have a skin cancer, and specifically basal cell carcinoma does that a lot. You need to have these surgically removed because they just spread and get bigger and causes more disfigurement. Squamous cell carcinoma is the second most common type, and it can metastasize. So it involves the keratinocytes of the stratum spinosum, and it's usually the scaly reddened papule on the scalp, the ears, the hands, the lips, things like that. Good prognosis if you treat early by radiation therapy or if you remove them surgically. And this is what that squamous cell carcinoma looks like. So you see it looks very different from the basal cell carcinoma we just saw, but radiation and or removal by cutting it out is a methodology to get rid of it. Melanoma, of course, that's the cancer of the melanocytes. It is the most dangerous type because it is highly metastatic and it is resistant to chemotherapy. So it's treated by a wide surgical excision accompanied by immunotherapy. So the key survival is early detection, and we use this ABCD rule. And so you can use ABCD rule to look at the blemish or the place on your skin, and is it asymmetry, or the two sides, the pigmented in the areas do not match? Do they have border irregularity for B that exhibits indentations? For color is C, it contains several colors. It could be black, brown, tan, sometimes red or blue. And then, of course, we look at the diameter for D. And if it's larger than 6 millimeter size, think the size of the pencil eraser, and that will kind of tell you if that's too large. Here's an image of that melanoma. So you can kind of see that this blackened area, you can see it's discolored. And, of course, it's quite a large place, and it can occur in various places. Kind of one of the last topics we'll talk about are burns. Tissue damages that are caused by heat, electricity, radiation, or certain chemicals, typically caused by the denaturation of proteins, that which destroy cells so that they no longer function. The immediate threat is dehydration and electrolyte imbalance. It can lead to renal shutdown and circulatory shock. So to evaluate burns, we use the rule of nines. So the body is broken down into 11 sections, each presenting 9% of the body surface. That's exception of genitals, which counts for 1%. So the rule of nines is important, and that helps estimate the volume of fluid loss. We also can classify them by severity. We have first-degree burns, which is localized redness, edema, which is just swelling and pain. And then second-degree burns, which is the epidermal and upperdermal damage, which you see blisters. We tend to refer to these as partial thickness burns because only the epidermis and upper dermis are involved. But the most severe, of course, is third-degree burns, and that's the entire thickness of the skin is involved. These are also called full-thickness burns. So the skin color turns gray, white, cherry red, or blackened, and no edema is seen, and the area is not painful because nerve endings are destroyed, and skin grafting is typically necessary. So for partial thickness and full thickness, you see here, first-degree burns, you see at the top here, this is a second-degree burn, and this, of course, is a third-degree burn. Now, in the last slide, it mentioned that in third-degree burns, you typically don't have pain. That does not mean that the person with a third-degree burn experiences no pain. It just means in this localized area, those specific nerve cells and nerves are damaged, and so those specifically aren't feeling pain, but they're going to have skin grafts, they're going to have the cells trying to regenerate, swelling, and all kinds of stuff that would lead to some pain involved. So I don't want to make it sound like the textbooks make it sound like there's no pain at all, but yeah, there is. It's just in that localized place you don't have anything. Burns are considered critical if the body is covered in more than 25% of second-degree burns, or more than 10% of third-degree, or if face, hands, or feet bear third-degree burns. And really, treatment has really 4 options here, and you can do basically you see a lot of them done all the time. Debridement, which is just removal of that burned skin. Antibiotics, because infections are a problem, especially in third-degree burns. One of the common infections that people face with third-degree burns is the bacteria Pseudomonas aeruginosa, and it releases this green pigment. So if you see someone with a burn and they have a bandage over it and it starts looking green and you smell this sickly sweet smell, they're infected with Pseudomonas aeruginosa and they need to be treated. So they're usually treated with antibiotics, temporary coverings, and skin grafts, like I mentioned before. And, of course, we talked about just a second ago that rule of nines and the severity of burns, but we divide basically the body into different regions, and 11 regions that cover 9% of the body, so to speak. And you can kind of see, and then, of course, the genitalia is about 1% that give us the 100% of the total body. So that's how we kind of look at the severity of these burns. So that's a little bit about Chapter 5. If you have questions, first make sure to utilize all the material in the module. There's videos, there's notes, there's lectures, there's extra stuff there to help you, extra activities, but also feel free to reach out to me. I'm here to help, and I'm happy to help. So if you have questions, just please reach out. And I hope everybody has a great day. See you in the next chapter.